my name is Madlen Hubert and I'm an associate professor at the Department of Pharmacy at Uppsala University. And I would like to explain a little bit the systems that we work with in our group. So in drug development, when you're looking into developing new therapies, they need to be tested in simplified models and in vitro models before they can be tested in humans, which is then happening in clinical trials. Especially if you're looking into treating a gastrointestinal disease, such as Crohn's disease, you use at the moment very simplified models. These are based on very simple cell lines, so they do not really sort of mimic the human complicated intestine. And that is where our research comes in. So we are working with developing more human relevant models, and we call them human intestinal organoids. So how do we make them? So we actually get samples, tissues from patients. These can be from biopsies or endoscopies, and this is all done with patient consent. We then isolate stem cells from those tissues, and from those stem cells we can actually grow what we call mini guts or mini intestines that we can then use in our research. We can grow those organoids in a configuration that we actually have the side that takes up drugs or nutritions, so when you eat something in the gut. So it's facing the outside of our 3D intestinal organoids. So that is really easy for us then to expose them to different drugs, for instance, and we can see how they affect the organoids. So we, at this point, have fully characterized those systems, and we have them also from relevant tissues, such as the ileum within GENEGUT. And we're now testing nanoparticles that are loaded with RNA therapies that are being developed within the consortia in these organoid systems. So the goal of our research is really to develop reliable and robust human relevant systems that we can use to predict the outcome of those therapies in patients much better. And then this is especially with a focus on hard to treat diseases such as Crohn's disease.